After alignment, we need to convert the larger size SAM file into smaller size BAM file. And for this step, I will use an example of real data. So let me look at where am I now. Okay, it's in Ebola folder. And that is the BOTA and the BWA SAM file we generated. But this time I want to use a different data set, this SRR FastQ file. Let me give the SRR variable. And then when I do BWA mem alignment, I want to add read group information, because later on I plan to use this file for variant calling and the GATK process. And for variant call, they're gonna need this RG read group information. I put it the SRR, the run sequence run number into this read group ID, and also sample ID, and also this LB tag. And the reference, same as before, the Ebola genome virus reference file, and the first FastQ file, the second FastQ file, and the output, I plan to put it into the BAM folder, and the name is the same as the sequence run number. Okay. Now let me convert the SAM to BAM file. Go to the BAM folder. The SAM file has a size 65 MB, and I use the SAM2's view. I think the newer version SAM2 has a more convenient command to combine those steps together, like the sorting and the indexing. But I used to do it step by step. This step convert SAM to BAM. And the BAM file is only 1/3 of the size of original SAM file. Next is SAM2's sort command. Sort the BAM file, and the output is sorted. I will call it sorted BAM file. It will be smaller after sorting, only 13 MB. Usually we'll delete the SAM file and only work on the BAM file. And the last step after sorting is index sorted BAM file. So with the index, we can view it in IGV or other genome browser. Okay, next I will give a brief introduction to variant calling. Variant calling is the process of identifying and categorizing the differences between the measured sequence and a reference genome. So here the word reference make it sounds like we are comparing our sequence read against the ground truth, but actually it's the other way around. It's our sequenced data that really exists, and the reference genome is often not even a real sequence. The reference genome does not represent a genome of any single person. Instead, it's a mosaic of different DNA sequences from each donor. For example, GRCH37 is derived from 13 volunteers from Buffalo, New York. And it's also very likely that this reference genome is not even viable. That means if those sequences were implanted into a human cell, the human cell would not function. Very likely would not function. So now think about it for a second. Scientists are using a non-existing, potentially non-viable human reference to characterize all other real human genomes. And when we compare two real human genomes, we do comparison by first comparing them via the third-party non-real one, and then comparing the results. So I think it's useful to think of the reference genome as a mirror, and we use this mirror to reflect our real data and make the data visible to us. It's an imperfect mirror, but it's a useful one. After all, the reference genome also changes over time. Then will a variant detected from the previous version of a genome still be a variant in the updated genome? So when we're talking about the variants, always specify which reference we are comparing to. Okay, so at the simplest level, variants are usually determined from alignment files, and the typical process involves the following 5 steps: alignment, correction and realignment, variant call, filtering, and annotation. Each of these 5 steps allow for substantial customization, and those customizations will in turn result in different outcomes in the last step. So what are the most commonly used generic variant callers? BCF tools, GATK, Freebase, and var scans. There are also specialized softwares that were designed for different applications, such as somatic mutations, germline mutations, family trials, or many others. And the genomic variation are typically categorized into different classes. SNP is a single nucleotide polymorphism, or a single base change. More strictly speaking, there is a frequency requirement. This single base change has to be occurring commonly within a population, for example, greater than 1% or 5%. But often, this population-wise data, the frequency in a population, is unknown. So when we see we have a SNP, we just infer that we observe a single base change in this particular sample. Indel is an insertion or deletion, is a single base addition or remove. SNP is a single nucleotide variant. It's either a SNP or an indel. MNP is a multi-nucleotide polymorphism. It's several consecutive SNPs in a block. MNV is a multi-nucleotide variant. It's multiple SNPs and indels as a block. Short variation that are typically less than 50 base pair in length. Large-scale variation is usually greater than 50 base pair. Structural variation is a variance that on kilobase scale. As a rule, SNP are the most reliable detectable variants from the data, and in contrast, very short MNVs are the most difficult one to detect. These include insertions and deletions. For example, a deletion followed by another variation can often be better aligned in alternate positions in the genome, and that can fool the variant callers. And for each class of genomic variation, different techniques are used. For example, for short variations, individual short reads can capture them entirely, and the large-scale variants are typically detected from the relative position of the reads about their read pairs. And for structural variants, long reads are usually used. So in the variant calling process, we'll run into some terminologies that are not always used consistently. So we'll attempt to define a few of these terms, but please note that those definitions are not necessarily textbook definitions designed to accurately capture the proper terminology of genetics as science, but our definition is more like what people call certain features of their data. So first, the ploidy is the number of complete sets of chromosomes in a cell and hence represent the number of possible alleles of the DNA of a single cell. So this can directly correlate with how many possible alleles we can see in our sample. If all the cells carry the same DNA, then the number of alleles will correspond to the ploidy of the organism. If the cells represent a population of, for example, n distinct individuals, then the maximum possible allele we can see will be n times the ploidy of the organism. And a genotype is the genetic constitution of an individual organism. And in the context of genomic variation, a genotype typically refers to identifying one or more of the sequence under investigation. And the simplest way to think about a genotype is a collection of all known alleles. And we use the word genotyping. It means we can assign one or more sequence reads to a known genotype. A haplotype is a group of genes in an organism that is inherited together from a single parent. In the context of genomic variation, a haplotype is a group of mutations that are inherited together. Before we dive deeper into the variant calling, it's important to understand that all the variant detection operates on alignment files. Even though the so-called variant callers can refine alignments, they can only improve based on the existing alignments. Therefore, producing the right alignments to start with is essential. A pile-up is a data representation that shows the base composition at a given coordinate. A SNP caller operates on this type of data, a consensus over a coordinate or a region in the genome. Okay, now let's do a pile-up with a reference genome. Same tools, mpile-up. And this is the reference genome in the reference directory. Now is our sorted BAM file in the BAM directory. I don't want to show it. It's gonna be a huge file, so I'll use a pap it to a less command, so it will show up here. This produces the output. The first column is the sequence ID. Second column is the position in the sequence. It starts from 1. The third column is the reference nucleotide at that particular position. The next column is the number of aligned reads cover this position, or we can say the depth of the coverage. It's usually in the two ends of the sequence, it usually has less coverage. If you page down, go to the middle of the sequence, you can see there are increase in the number of reads to 300, 400. And the next column, it represents the base match at that position from the aligned reference. For example, the comma means that base is matched in the reference on the reverse strand. And if we see a dot, I haven't seen it here, here, the period, the dot means a base that matches the reference on the forward strand. And this capital T or AGTC means this base does not match the reference on the forward strand. Instead, in the reference is a G, instead in our read is a T. And the lowercase AGTC means this base does not match the reference on the reverse strand. So on the, for example, in this case, in the reference is a G, and in our read there is a insertion T and C. And the plus sign means there is an insertion, and the minus sign means there is a deletion. And this caret marks the start of the read. That means there is a read start from here. And if we see a dollar sign, it marks the end of the read. I didn't see a dollar sign here. Oh, here is a dollar sign. And the star, let me see. Okay, I didn't see a star here, but anyway, if we see a star, it means it's a placeholder for a deleted base in a multiple base pair deletion. And you can also try to pair up without a reference genome. So the only difference with Lac's previous command is this: without the reference genome, we can see the read sequence within our read instead of the comparison with the reference genome. We can also pair up, take location coordinates. For example, we want to look at the position 470. Now there are 770 reads aligned in that position. And out of the 770 reads, if we just look at what reads are aligned to the reverse strand, we can add a SAM flag. RF16. That is the reads on the reverse strand. So there are only 318 reads on the reverse strand.